So I'm going to talk to you about, we are a poxviral vector-based laboratory, and so most of the vaccines we work on are poxviral vector-based vaccines. And I'm talking about a vaccine, vaccine for HIV that we are designing. So I'll just give you a brief overview of HIV first and then go from there. So HIV, almost 30 years have passed since the discovery of the virus, and when the virus was discovered, it was said that we'll find a vaccine in 2 years. And still we don't have a vaccine, except the vaccine currently that is looking some promise is the RV14 trial, which is showing 31% efficacy. So HIV is a single-stranded RNA virus transmitted as an envelope virus, and the envelope structure is giving us problems designing good responses. So basically, HIV infects CD4 T cells, and the biggest problem is HIV, basically, once it enters, it can basically get integrated into your genome, and that's where the problem begins. And it can stay dormant for a long time, and then it starts to bud out, and that's where the problem, HIV latency is the problem. So basically, when it comes to HIV, your CD4 T cell numbers will decline, and HIV, it becomes AIDS when it goes below 200 counts, CD4 counts. And I basically work as a volunteer in South Africa, and until very recently, the kids, especially many people who suffered from HIV, they were only treated with intraretrovirus when they went below 200. So it's a big problem in places like Africa. So basically, currently there's about 35 million people are living with HIV/AIDS. 2.4 million are children, and there are quite a lot of orphans. And 2012, there were about 1.6 million deaths and 2.7 million new infections. And there's 10 infections every minute, and we don't think about it. And 95% of the new infections are in the developing countries, and Africa is the biggest problem. And currently, India, Thailand, China, it's becoming a problem as well. And even in Australia, I think people are not basically taking, the younger generation, they are not taking precautions, so the HIV is up on the rise at the moment. So, and it's really devastating to see if you visit Africa and in an audience like this, 80% of the people will be HIV positive. And it is unimaginable when you live in a country like Australia. So basically, HIV, there are different clades according to the different regions. And so basically in Australia, we have B clade, but in Africa, you have C, and then in Thailand, there's AE clade. So the clades are another problem. That's another problem why we can't design a universal vaccine. So people might think that there's a cure. There's no cure for HIV. Antiretroviral treatment is not a cure. It is just, it's keeping your virus at bay. And if you get out of retrovirals, you get your virus back. So there's no cure for HIV. Currently, the one that is looking very promising antiretroviral drug is Truvada. So basically, still we need a vaccine. So the biggest pitfalls we have had in HIV vaccines is that we have had very good promising results in animal models, but when we have gone into human trials, they have failed. But the thing is that all the vaccines we have trialed to date, they have been systemic vaccines. We are delivering to the bloodstream. But HIV is not disease of the bloodstream, it's a disease of the mucosa. So the many trials we have had is DNA prime boost trials, one of the trials that even I was part of a few years back, about 10 years back. That was DNA prime followed by a recombinant Falpox booster. And the next step trial was the one that they thought the magic bullet in 2007 and failed miserably. And the one that is looking promising at the moment is a Thai RV14 trial. That's 31% efficacy, and that's a canary pox prime followed by a protein booster. But the thing is that the vaccine is only generating antibody responses to envelope. There's no T-cell immunity. So what people are thinking is that if you add the T-cell arm to this, the vaccine might be successful. So as the vaccines are failing, and no one wants to understand why the vaccines are failing. What are the correlates of protective immunity? 10 years ago, we did not know that. Currently, we have some idea. Then we need to develop better vaccines. There's no point doing systemic vaccines if they are not working for a mucosal disease. It's like mucosal vaccination. And also, if we find a vaccine, basically we need to also find that how are these vaccines working? Because last trial in Sydney trial, $26 million later, the vaccine failed. And so that's much it is cost for a vaccine. And people basically, we need to take vaccines. We have to be certain, have some guarantee that the vaccines are working before we go and do a trial, because trials are very costly. So basically, why a mucosal vaccine for HIV? It infects basically, the first encounter is the genitorectal tract. The CD4 CD depletion that happens at the very beginning happens at the gut mucosa. And also, therefore, immunity at these sites are extremely important. There is no point giving blood immunity if you don't have mucosal immunity. And you cannot generate mucosal immunity, as Alan yesterday mentioned. You cannot generate good mucosal immunity if you give a systemic vaccination. You need to give a vaccine to the mucosa. So basically what we are finding with the poxviral system, if you are using poxvirus vaccines, what you are finding is that if you give a vaccine, this is generally true with many vaccines, but I'm talking about poxvirus vaccines here. What we are finding is that if you give a vaccine intranasally, we are generating immunity in the upper respiratory tract, the gut, the genitorectal tract, and the mucosa compartment at the genitorectal mucosa. And if you basically give it orally, we generate immunity in the in the gut and even the mammary gland. Intrarectally, the gut and the rectal mucosa, and intravaginally, we are seeing in the vaginal mucosa. So again, you can see that if you give different routes, the immunity you are generating is quite different. So when it comes to HIV, therefore intranasal vaccination is quite important because it's generating immunity at all compartments. So for HIV, we are looking at this about 9 genes that people are interested, and mainly we are interested in the GAG, pole, and envelope. That means GAG and the pole are internal proteins that are generating T-cell immunity, and envelope is the outer membrane protein that is generating antibody responses mainly. So what we have done is, as I said, we work on poxviruses. We have recombinant fowl poxvirus. It's basically a virus that basically grows in chickens, but it doesn't infect. It will infect people, but it doesn't replicate in humans. So it's an abortive virus. It's similar to canary pox, where it's the same family. And we basically have engineered our HIV GAG pole envelope genes. And our second virus is basically our recombinant modified vaccinia ankara or vaccinia. We have used vaccinia, but as we are going human trials, we are going into modified vaccinia ankara. Again, a non-replicative virus, and we have basically included HIV GAG pole. So what we have done is, as the vaccines were failing, what I went and asked was, what if we give the vaccination different routes? What do we get outcomes? And from there onwards, can we find what is why the vaccines are working or not working? So we have gone on a mission where you have purely mucosal systemic immunization, purely mucosal immunization, and a combined mucosal systemic strategy. So you prime with fowlpox. Fowlpox is an excellent mucosal delivery vector. So if you basically engineer your virus, and if you give it intranasally, it just gets taken up very well. And the booster should be modified vaccinia ankara or vaccinia. So if you go and look at the magnitude, all the people, everyone in a vaccine trial, we seem to be looking at magnitude. So if you look at magnitude of response, everyone say systemic vaccination, magnitude is great. But if you look at the qualitative differences of T-cell responses, the quality of a systemic vaccine is extremely poor. and quality of a mucosal vaccine is much better. Then it's quite good. So basically, intranasal Falpox prime followed by intramuscular modified vaccine and Cara boost is generating very good quality, magnitude as well as quality responses. And the question was, why is this? What is the reason why these vaccines are not good quality? So what is important, magnitude or the quality? And we think that all the HIV vaccines are failing because we have been looking at magnitude. Because systemic vaccination is great, we go and we fail. And so we ask the question, why is this failing? And cut a very long story short, what we are finding is that if you give a systemic vaccination, we are generating T cells that are poor quality. And the reason is that these T cells are producing IL-4 and IL-13. But if you give us mucosal vaccination, you are generating T cells that are producing IL-4, IL-13, but they are lower. The numbers are lower. So what we did was, I was lucky to be at the John Curtin Medical School where we had the IL-4, IL-13 knockout animals. And if you basically vaccinated mice that were IL-13 knocked out, what we found was we were generating extremely good quality T cells. I really didn't believe the results for about a year. And then basically, IL-4 is the same, but out of the two, what we are finding is that the IL-13 knockout animals were behaving much better than the IL-4 knockout animals. So then we said, okay, we can't create knockout humans, so we need to go and find whether we can find a vaccine that we can block IL-4, IL-13. So this is where what we did was, we took our vaccines, we basically integrated two vaccines. One vaccine, we introduced a soluble IL-4 receptor alpha-2, and the other one, we introduced an IL-4 receptor antagonist. And we created these into both vaccines. So this is how these vaccines work. Basically, IL-13 receptor alpha-2 adjuvanted vaccine, basically what it does is, once it's given intranasally, it will basically express your antigens, and also it will basically start to express your IL-13 receptor alpha-2. It will sequester the IL-13. It will basically mop up the IL-13 in the vaccination site. So basically you are reducing the amount of IL-13. And the other vaccine, we are blocking the IL-4 receptor pathway. It is basically preventing temporary IL-4, IL-13 signaling. Only this pathway is available, and so our two vaccines are working by blocking two pathways. We are mopping up IL-4, one is mopping up IL-13, other one is reducing the signaling via these two pathways. So as I said, we need to know these vaccines are safe and how long these are expressed. What we are finding is that our vaccines basically starts to express within the first 6 hours as soon as they get in. At 12 hours, you have peak expression. 48 hours, 96 hours later, you don't see anything. So the vaccine only lasts in the system for 96 hours. So the virus, it's an abortive replicative virus, so once its antigens are expressed, it's gone. So it's safe. So we wanted to check whether, as you're putting down the nose, we want to see whether they are safe. So we expressed MCHERI in the virus, and we looked at in lung. You can see the expression in lung. Again, 96 hours later, you don't see anything, but it doesn't cross the blood-brain barrier. You don't see anything in the brain. This is some live animal imaging. Again, you can see control animal, immunized animal. You can see it's basically expressing only in the lung. So as I said, we wanted to know the quality of responses. Again, what you're finding is that if you give this vaccine in the prime and the booster, these IL-13 knockout animals, you are generating good quality T cells. And again, if you only give it in the prime, we generate good quality T cells, but if you give it only in the booster, we don't create any good quality T cells. So this suggests that inclusion of the prime inhibitor in the priming vaccination is important, because if you don't create the first T cell repertoire that is important, that high quality, you can't create that repertoire ever again. So what we are finding is that these vaccines, what is happening is that within the first 24 hours, we are creating a repertoire that is good or bad. So there's no point messing around with the vaccine afterwards, trying to make it better if you have not made that change at the very beginning. So what we are finding is, yes, if you give it in the booster only, you get quite a lot of magnitude, which was poor quality, but in the prime and the booster, you are getting very good magnitude. So basically what you are finding is that out of these CD8 T cells, like 20% or 14% of our CD8 T cells are producing interferon gamma, which is pretty good. And this is basically systemic immunity, so adjuvanted vaccine, unadjuvanted vaccine, but I'm more interested in the mucosal responses. And as you can see, we are generating excellent mucosal immunity in the lung, the genitorectal compartment, and the gut compartment. And we have not had a vaccine that basically generated immunity in all compartments. So this is, again, a parameter that you have to look at for quality of responses. Again, you need to look at multifunctional T cells. And if you look at multifunctionality, this is our new vaccine. This is our control vaccination. You can see that we are producing multifunctional T cells, interferon gamma, and TNF alpha. So in all compartments again. So this is a vaccine that went into trial in Sydney 10 years ago. These are the positive controls. This is a 64 cytokine array. You can see this is the interferon gamma level we found. And this was the best vaccine that we had. And everybody in the world, they took with these vaccines trial, that was what we found. This is our control vaccine that has not gone into trial, intranasal intramuscular vaccination. This is our new vaccination, and this is the interferon gamma level we are producing, and it is lighting up every cytokine, chemokine that we should basically see in a good vaccination. So this is lots of chemokines, cytokines, everything that you see in a good vaccine is lighting up in our vaccine. So when we looked at the protection level of these vaccines, the two vaccines, what we're finding is that we can't give HIV to mice, so we had to be a cheat. We had to create a flu virus expressing HIV epitopes to do this. So we have created a flu virus. So if you give, these are control animals just given the flu infection. This is our control vaccine, which is not bad. These are new vaccines, they're protected. So what you're finding is that our vaccines are generating good, high quality T cells that are generating good T cell mediated protective efficacy. So what about antibodies? You can't have one thing, we need to have the other arm. So basically, what we are finding is that our vaccines are able to generate good antibody responses, IgG1, IgG2A. And what we are finding is that basically our second vaccine, IL-4 receptor antagonist vaccine, we are having antibody class switching that is happening compared to the IL-13 receptor alpha-2 vaccine. And antibody class switching currently, like 2 years ago, it was shown that in people who control HIV naturally had antibody class switching. And also people who control HIV naturally have good quality T cells. So our vaccines have the two parameters that a good vaccine is we want to generate, because these parameters are not known when we started the work. So then we said the Thai trial basically have the booster arm where you are generating envelope responses. So we said if we give a protein booster, can we generate also envelope responses, like similar to the RB14 trial that is successful? And what we are finding is, of course, yes, our vaccines are generating good antibody responses, IgG. These are endpoint titers, and basically we are generating good IgG1 antibodies as well. But also we are finding is that if you do a qualitative assay using guanidinium thiocyanate to dissociate antibody responses, what you're finding is that even though we don't see with this vaccination at the beginning good quality T cells, at the end, basically they catch up and we generate good quality T cells. And this is due to the two vaccines are using two different pathways to induce antibody responses, which I don't have time to talk about, and that's the reason why we are basically generating these responses. So how do these vaccines work? What we are finding is that, as I said, it happens at the very first 24 hours of vaccination. The intranasal vaccination, basically what we are finding is that Fowlpox priming is essential, because Firefox is a virus basically generating, basically what is happening is that this is our control vaccine, this is IL-13 knockout animals, this is the alpha-2 vaccination, this is the IL-4 receptor antagonist. What you can see is we are generating high amounts of MHC positive, CD11C positive, CD11B positive, CD103 negative T cells here. Okay? So what is happening is that we are basically, even though people say CD103 is important and generating cross-presentation, what we are finding is that at the very beginning, cross-presentation is not a good idea, because cross-presentation generates poor quality T cells. So if you gave a prime with MVA, for example, modified vaccine NCAR, this will stop completely. What happens is you are generating quite a lot of CD103 positive populations here, and then that you are generating poor quality T cells. So Firefox priming is pretty essential, and our antagonists are very important to be there because they are generating quite a lot of CD11B positive cells. And these CD11B positive, CD11-3 negative cells are very important because we have done transfer studies and we have shown that they are the ones who are playing a very important role in generating quality of T cells. And so also if you look at basically CD B220 positive populations, you are seeing a difference between these two, this versus that, and we think that that population is playing a role in B cell immunity. And at this moment we are looking at that aspect in the lab. And also I would like to just mention that I don't have time to show the data. What you're finding is that who are the things that are producing IL-13 at the vaccination site? Everyone asked me the question. And it is the newly discovered inert lymphocytic T cells. ILC2s are the major source of IL-13 at the vaccination site, which is basically generating quite a lot of IL-13 in the lung mucosa. And if you mop it up, you can basically generate better quality T cells. So, and that's what we are finding. And so what we are finding at the moment is that we are generating a vaccine that is generating very good quality T cells. And high quality T cells are important in HIV natural controllers. We are generating antibody class switching. Antibody class switching 2 years ago was found, GAD specific antibody class switching is involved in basically HIV control. Good envelope specific antibodies, that's the only thing that we have found in the RV14 trial that is protective. So we have 3 factors that are currently showing that these vaccines basically are, we have so-called a vaccine that is basically generating triple action immunity, where we have good T cells, GAD-specific antibody class switching, and envelope-specific responses. So we think that this strategy is basically not only for HIV, we can use it for other diseases, mucosal diseases, and currently we are doing some work with Ken, a vaccine on chlamydia, and I have some collaborations on TB as well. And so we are also currently thinking of combining our vaccine with the RV14 trial so that we can give the arm that is missing from the RV14 trial, and we are having some collaborations going on. And these vaccines currently are we are started the macaque trial currently in Melbourne. So we are at the stage of translating the vaccine to preclinical studies. So basically I didn't do my work all by myself. There's a team behind everything, and Ron Jackson, who is basically a pox guru, who is in my laboratory and who has amazing pox virus knowledge, and he's the one who is behind creating the vaccines. And there's lots of students and staff who have worked in this, and also my collaborators, Rob Center, who is in Melbourne, who has provided me all the protein, and David Boyle, again the pox virus guru in Australia, who has given me all the pox virus constructs, and Alistair, who is at Louisiana Vaccine Center, whom I'm having some collaborators, and all my collaborators in Melbourne, and my funding bodies. Thank you.
Time for a couple of quick questions. Okay.
So it's a SIV strain of gels, the macaque.
This one, the macaque is a SIV trial, so MAC 239. And we are also planning an HIV trial, and we would like to go SHIV, but there's another replicative virus where you can use the one that is having everything. So if I can get hold of that, that's what I would like to use. Okay, for mouse studies, yes, we do a 2-week, but longer you leave it, the better it is. So basically what we are finding is that we have done some studies, we have left the mice for like quite old, like 6 months later, and you get massive responses. And what we are finding is that that's what I was told by people who are doing the RV14 trial. And because RV14 trial, you are doing 2 or 3 canary pox primes, followed by several protein boosters. And what they are finding is that if you basically give the boosters far apart, you are generating better immunity. So in a human trial, I think that would be good if you can start the prime and then basically 1 intranasal prime, leave for a long time, come with the booster, that might be the way to go.
Okay, in the interest of time, I think we've got to